Okay, hello, welcome to this video from Mr. Gill's Science Stuff. We're talking about how you prevent and how you would calculate bacterial growth today, so how quickly they grow and how you would stop them doing so if that's what you wanted to. This is part of the pathogens and disease topic in the GCSE Biology. So what we're going to do is we're going to look at firstly how bacteria multiply, how not to confuse them with our own cells, calculating the number of bacteria in a population, and then lastly how we stop them from growing, because a lot of the time that's what we want to do. So the specification looks like this. There's some keywords there like binary fission, a little bit on their growth rate and what conditions help them to grow and what stop them to grow, and then a little bit on a very useful piece of science equipment, biology equipment, the agar gel plate at the end. So you should know already what should be underneath all these little signs here, or these little boxes here, so what the parts of a prokaryotic cell are. So if you want to take a moment to pause the video at this point and have a little go at working out what should be under 1, 2, 3, 4 and 5. Okay, done that? Right, what you should have got is that under number 1, that's the genetic material. Remember, that's how you tell a prokaryote from a eukaryote. You've got a loose, no nucleus, single circular chromosome. Number 2 is the cytoplasm, just the same as our other cells. Number 3, the cell membrane, again just the same, controls entry and exit from the cell. You've got the cell wall out here, same function but made of different stuff. Remember, it's not cellulose, it's either peptidoglycan or murine. And number 5, we've got plasmids. Okay, so those are our extra little bits of DNA. Now the other things on there, the slime capsule and the flagella, you may have learned all about, and that is correct, they are there quite a lot of the time, but they're not on every single bacteria, so that's why they're not under one of those boxes. So moving on quickly, new keyword for you here, binary fission. Okay, so this is very, very simple, very basic cell division. Bacterial cells much smaller than your cells, much more simple, much less DNA. So they can literally replicate their DNA, right, to make a new loop of DNA and then split the cell in half every 20 minutes, and that's 2 new bacteria. Okay, so long as they've got plenty of nutrients, plenty of oxygen, plenty of water, plenty of the materials to build their molecules from, they will very, very happily just keep multiplying once every 20 minutes. Now the key word here, this is called binary fission, not to be confused, GCSE students, not to be confused with mitosis. Okay, now mitosis is your cells, that's where our chromosomes all line up and get pulled apart and so on and so forth. Now I've seen A-level students and even higher level students making that mistake before. This is binary fission. Mitosis is about eukaryotic chromosomes lining up and getting pulled apart and all that malarkey. This is binary fission. Okay, so make sure that keyword is written down somewhere and you learn it, that when we're talking about bacteria, it's binary fission. Okay, now obviously that doubling rate, now if you're quite good at your maths already, you'll be seeing this explains that they grow pretty quickly. Okay, so if a population in ideal conditions can double every 20 minutes, before too long you're going to have a huge quantity of bacteria. And that is exactly what happens. Okay, in ideal conditions, bacteria doubling every 20 minutes, if you work it out from the mass of a single bacteria, even one bacteria being very, very small, one bacterium being very tiny, if it was to be able to double every 20 minutes, you'd have a ball of bacteria larger than the Earth in under 48 hours. Okay, so that's a incredibly rapid growth. And if you don't believe me, you can check the maths on that. So obviously that doesn't happen because they don't have the right conditions and they don't have the right nutrients to be able to grow. Right? If it were bigger than the Earth, what would they be growing into? But you have to be able to calculate what number of bacteria you would have after a certain amount of time given ideal conditions. So a little bit of maths for you in this lesson, and we're going to do a couple of examples together. So here's the first one. So the strategy goes like this, right? You look for the number of bacteria in the initial population. Often you're given it or you have to count it off a plate, okay, off a picture of a plate. And at the end, you'll have the number at the beginning times by 2, because obviously whenever they divide in 2, multiplied up by the number of divisions. So if they've only been going 20 minutes and you started with 4, you'll have 8. If then you gave them another division, you'd go from 8 to 16. 16 to 32. So the expression here is number at the end equals number at the beginning times 2 to the power of the number of divisions, so the amount of times they've been allowed to divide and multiply. So here's an example to make that clearer. If one single bacterium landed on a cake that you might be eating at the right temperature, how many will there be after 2 hours if they reproduce it every 20 minutes? Okay, so in 2 hours they would have reproduced 6 times, because you've got 3 lots of 20 minutes in every hour. 2 hours, that's 6 multiplications. So the number of divisions they've been through is 6. You started with 1, right? You've started with 1, so that's 1 times 2 to the power 6. Okay, because you can work it through. If you've got a small number here, after 20 minutes you'd have 2, after 40 minutes 4, 60 minutes 8, 80 minutes 16, 100 minutes is 32, 120, 64, so on and so forth. Okay, so you end up with 64 that way. So the number at the end is the number at the beginning times 2 to the power of the number of divisions. So that's 1 times 2 to the power 6 equals 64. Okay, so we're going to do a few questions together. Right, we're going to work through the answers to these. So I want to show you an important button on your calculator, which means I now need to bring in my second webcam, which is down here. Okay, now on your calculator, presumably you've got the same lovely standard calculator that I've got. You've got this button just here, okay, where it says X with a little box up at the top. Right, now if I wanted to do that calculation we've just done, 1 bacteria times 2, right, to the power of 6, so it's been going 6 generations. Now you can close the bracket or not, it doesn't make a big difference here in this particular type of calculation, equals 64. Okay, so that's how you do these examples. So I'm going to put the questions up. I'll take that away for a minute, bring it back in a moment. So here's our questions. If a bacterium lands on a cake at the right temperature, how many would there be after 5 hours if they multiply every 20 minutes? If 40 bacteria landed on a cake at the right temperature, how many would there be after 4 hours if they multiply every 20 minutes? And lastly, if 100 bacteria landed on a cake at the right temperature, how many would there be after 6 hours if they multiply every 30 minutes? Okay, so we're going to stop this part of the video here, right? And part 2 will start with the answers. So pause here, keep those on your screen, have a go and see if you can work that out, and I'll give you the answers at the beginning of the next video. Okay, thanks.